Well, I've started wearing sweaters, sipping hot beverages, and lighting candles. So I guess it must be fall. And if it's a new season, there must be a new breakthrough, a new promising study related to multiple sclerosis. And lo and behold, there is. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam and I've been living with multiple sclerosis for 38 and a half years. I have been following all new developments that I can come across and trying to evaluate them a little bit, but certainly to share them with you so that you can evaluate them too. And I have a new one for you today. It's quite exciting, I think, if it would really work. So much of what we hear, we have to take with a dose of either skepticism or at least prudence and patience, because even if something does turn out to be the perfect thing, the most wonderful thing ever, it isn't going to be ready for prime time anytime soon. But, oh well, we might as well at least know about it, because if we're going to be seeing our doctors soon, we could ask them about it. And wouldn't that be helpful to know whether our doctors are keeping up with the latest and greatest in multiple sclerosis research and the new studies that are coming out? So let's go ahead and take a look at what I found. Well, here's the article, and it comes out of the University of Chicago. It looks like it's published right on their website as opposed to coming out of a news media outlet. So the Pritzker School of Molecular Engineering at the University of Chicago has recently published, on September 11th, 2023, in fact, an article, Inverse Vaccine Shows Potential to Treat Multiple Sclerosis and Other Autoimmune Diseases. Scrolling down to the beginning of the article, it says, a new type of vaccine developed by researchers at the University of Chicago's Pritzker School of Molecular Engineering has shown in the lab setting that it can completely reverse autoimmune diseases like multiple sclerosis and type 1 diabetes, all without shutting down the rest of the immune system. Now, this is an extraordinary claim, and if there's any actual merit to it out in the actual world as opposed to in the laboratory, we are talking about a very important breakthrough. They go on to say, a typical vaccine teaches the human immune system to recognize a virus or bacteria as an enemy that should be attacked. The new inverse vaccine does just the opposite. It removes the immune system's memory of one molecule. While such immune memory erasure would be unwanted for infectious diseases, it can stop autoimmune reactions like those seen in multiple sclerosis, type 1 diabetes, or rheumatoid arthritis, in which the immune system attacks a person's healthy tissues. And they continue, the inverse vaccine described in Nature Biomedical Engineering, so in other words, it's been published in a peer-reviewed journal, that's good to know, takes advantage of how the liver naturally marks molecules from broken down cells with do not attack flags to prevent autoimmune reactions to cells that die by natural processes. PME researchers coupled an antigen, a molecule being attacked by the immune system, with a molecule resembling a fragment of an aged cell that the liver would recognize as a friend rather than a foe. The team showed how the vaccine could successfully stop the autoimmune reaction associated with a multiple sclerosis-like disease. So in a way, what they're doing here is they're linking this molecule, this antigen, to a molecule that the body would recognize as, oh, this is no threat. This, in fact, is going to be a helpful thing. It's a little bit like a Trojan horse, right? Except that in this case, it's for good and not for destruction. In the past, they say, we showed that we could use this approach to prevent autoimmunity, said Jeffrey Hubble, the Eugene Bell professor in tissue engineering and lead author of the new paper. But what is so exciting about this work is that we have shown that we can treat diseases like multiple sclerosis after there is already ongoing inflammation, which is more useful in a real-world context. And again, as I look at that, I'm thinking, okay, you're going to stop the smoldering MS, because I don't know how else you're going to reverse the inflammation that's already present. There's so much going down at the cellular level that if you don't have something that's going to stop that, you're really not going to be affecting any kind of long-term cure. And here's a photo of Dr. Jeffrey Hubble. He seems like a nice guy. He certainly seems happy with the progress that he's made. And I don't blame him at all. And they go on to say, unwinding an immune response. The job of the immune system's T cells is to recognize unwanted cells and molecules, from viruses and bacteria to cancers, as foreign to the body and to get rid of them. Once T cells launch an initial attack against an antigen, they retain a memory of the invader to eliminate it more quickly in the future. T cells can make mistakes, however, and don't we know it, those of us with MS, and recognize healthy cells as foreign. In people with multiple sclerosis, for instance, T cells mount an attack against myelin, the protective coating around nerves. Hubble and his colleagues knew that the body has a mechanism for ensuring that immune reactions don't occur in response to every damaged cell in the body, a phenomenon known as peripheral immune tolerance, which is carried out in the liver. They discovered in recent years that tagging molecules with a sugar known as N-acetylgalactosamine, PGAL, or PGAL maybe, I don't know, could mimic this process, sending the molecules to the liver where tolerance to them develops. The idea, they say, is that we can attach any molecule we want to PGAL, and it will teach the immune system to tolerate it, explained Hubble. Rather than rev up immunity as with a vaccine, we can tamp it down in a very specific way with an inverse vaccine. I don't know about you, but I just find this idea pretty fascinating. I don't know whether it's going to end up having legs, but it's really pretty fascinating because, again, it's a different approach, and there does seem to be some good science behind it. They go on to say, in this new study, the researchers focused on a multiple sclerosis-like disease in which the immune system attacks myelin, leading to weakness and numbness, loss of vision, and eventually mobility problems and paralysis. The team linked myelin proteins to PGAL and tested the effect of the new inverse vaccine. The immune system, they found, stopped attacking the myelin, allowing nerves to function correctly again and reversing symptoms of disease in animals. Now, I'm thinking this is the famous EAE, inducing mice to develop a neurological disease that's similar to MS. And we do know that there are some drawbacks to that. There are some limitations to that because we are not mice, and our bodies do not respond exactly the way the body of a mouse will respond, either to the disease or to any treatment. And they go on to say, in a series of other experiments, the scientists showed that the same approach worked to minimize other ongoing immune reactions. And then the last section here, toward clinical trials. Today, autoimmune diseases are generally treated with drugs that broadly shut down the immune system. These treatments can be very effective, but you're also blocking the immune responses necessary to fight off infections, and so there are a lot of side effects, said Hubble. If we could treat patients with an inverse vaccine instead, it could be much more specific and lead to fewer side effects. And wouldn't we all like to see that? Initial phase 1 safety trials of a glycosylation modified antigen therapy based on this preclinical work have already been carried out in people with celiac disease, an autoimmune disease that is associated with eating wheat, barley, and rye, and phase 1 safety trials are underway in multiple sclerosis. Those trials are conducted by the pharmaceutical company Inokion SA which helped fund the new work and which Hubble co-founded and is a consultant board member and equity holder. The Alper Family Foundation also helped fund the research. There are no clinically approved inverse vaccines yet, but we're incredibly excited about moving this technology forward, says Hubble. And now let's go ahead and click on this link, because I believe that's going to take us to the paper. Yes, there we are. That we're at the paper that was published, Nature Biomedical Engineering. It's called Synthetically Glycosylated Antigens for the Antigen-Specific Suppression of Established Immune Responses. And there really isn't any point in my going through this because without paying for it, I can only get the abstract. And we've already more or less covered all of this in the article that was written about it. But I will link to this in case you have access to the whole paper and you want to go ahead and read it. But I do think this is a pretty interesting and I would say novel way to go about combating MS. I really do like the idea that it might represent a reversal and a repair of much of the damage, especially if you're in the early phases of MS. Well, the University of Chicago is certainly a worthy institution, and great things come out of there all the time. This is probably the first time I've ever seen their name associated with multiple sclerosis, but it looks like the kind of work that they're doing could generate a lot more useful results than just for multiple sclerosis, as they say. But I also think it's a good idea if we could put this in context a little bit. And thankfully, I found another article from another highly reputable institution that will tell us in a little bit more detail what this research is all about and why it might be really important. Well, and it's neat to see that other prestigious institutions have taken notice of this new study. Here's an article out of the Massachusetts Institute of Technology Review. And it just came out September 22nd of 2023. The title of the article is How Inverse Vaccines Might Tackle Diseases Like Multiple Sclerosis. I do like how this starts out because the author expresses the same incredulity that perhaps many of us might have when we first see something like the idea of an inverse vaccine for multiple sclerosis. She says, I've written about vaccines for years, but recently I stumbled across a concept I had never heard of before. Typical vaccines prime the immune system to respond, but scientists are working on an inverse vaccine that teaches immune systems to stand down. Last week, Jeffrey Hubble and his colleagues at the University of Chicago reported that an inverse vaccine they developed had successfully reversed a disease similar to multiple sclerosis in mice. Hubble has tested this approach before, but only as a way of preventing the disease, not curing it. What is so exciting about this work is that we have shown that we can treat diseases like multiple sclerosis after there is already ongoing inflammation, which is more useful in a real-world context, he said in a press release. And yes, this would be very exciting, of course, because looking at not just preventing it, but helping those of us who already have it and have been dealing with it would be just wonderful. It would be a major, major breakthrough. As they say, these immune dampening shots could lead to a whole host of therapies to treat autoimmune diseases. In fact, Enochion, a company Hubble co-founded, has already launched clinical trials to test whether this type of inverse vaccine might help people with multiple sclerosis and celiac disease. It's an exciting prospect. So for the checkup this week, let's take a look at inverse vaccines. And this is good. How do these vaccines work? This is a question probably many of us have, so let's see what they say. Let's start with a little immunology 101. We tend to think of our immune system as a beefy bodyguard fighting off all pathogens that seek to harm us. But it has another equally important job. Mostly, our immune system ignores stuff that it's being exposed to all the time, says Megan Levings at the BC Children's Hospital Research Institute in Vancouver and a member of Anokion's Scientific Advisory Board. That includes all the food we eat, all the bacteria that live in our bodies, all the funguses and mold in the environment. The capacity to ignore, known as immune tolerance, isn't passive. The immune system learns which things are dangerous and which are not, and stores that memory in specialized cells. When the system makes a mistake and flags a harmless protein as dangerous, the mix-up can cause serious problems: allergies, autoimmune diseases, and other types of immune disorders. With traditional vaccines, the goal is to deliver a foreign substance in a way that raises alarms. That's why vaccines are often combined with ingredients called adjuvants, which provoke a stronger immune response. mRNA vaccines don't need adjuvants because the immune system already sees genetic material as a threat, while inverse vaccines, also called tolerogenic vaccines because they provoke tolerance, the goal is to train the immune system to recognize that a particular target is harmless. I should point out that the idea of tolerogenic vaccines is not new. Researchers have been working on them for decades, trying different methods for delivering the desired vaccine targets, called antigens, without provoking an immune response. But until now, they've had little success. Hubbell's group has developed a technique that involves adding a sugar to the antigen, which ensures that it travels to the liver. Why the liver? The organ has the ability to tag molecules with harmless labels. It's actually harnessing normal biology, Levings says. For a deeper dive into the paper, read Eric Topol's newsletter, Ground Truth. That's where I learned about the concept of inverse vaccines. And of course, I will link to this article and you can read that for yourself. But adding a sugar isn't the only way to develop an inverse vaccine. In 2021, a team from BioNTech and the Johannes Gutenberg University reported that they had developed a tolerogenic mRNA vaccine able to curb symptoms in several mouse models of multiple sclerosis. That's especially impressive given that mRNA tends to be so very good at prompting an immune response. The researchers achieved this by altering the fatty nanoparticle that carries the mRNA, but the exact mechanism wasn't totally clear even to Levings, who wrote a commentary on the paper. So often that's the case. Scientific breakthroughs, and technological ones for that matter, are often developed by people who haven't quite figured out why something works, only that it does. And it looks like the study into the mechanism is ongoing. Taking these therapies from bench to bedside won't be easy. It's tricky for a few different reasons, says Lawrence Steinman, a neuroimmunologist at Stanford University. First, with a complex disease such as multiple sclerosis, no single antigen is wholly responsible. So do you pick one, or do you want to make a complex mixture of many of those antigens, Steinman asks. There's also the challenge of proving that the vaccine works. The treatments for many autoimmune diseases have gotten much better over the years. About 15 years ago, Steinman led a clinical trial to test a tolerogenic DNA vaccine in patients with multiple sclerosis. The vaccine worked, but not better than cutting-edge therapies. We had a modest beneficial effect in reducing inflammation in the brain, but it could not compete with some of the drugs that were just coming on the market, he says. Now, Steinman serves as chairman for a company called Pasithea Therapeutics, and he's working on a new inverse DNA vaccine for multiple sclerosis. This one will target a protein in the brain that mimics a portion of the Epstein-Barr virus, which may be a trigger for MS. Now, there is something interesting as well. I'm glad to see that this is being developed on several fronts by different teams, because the chance of coming to something that actually works and gets approved and gets into the marketplace so that some of us can actually benefit from it It's much greater if it comes from multiple directions. Safety is another concern, especially the risk that these vaccines could provoke an immune response rather than dampening it, which would make matters worse. It's something that kept Steinman up at night when he was involved in that earlier trial. If researchers can get these vaccines to work, the payoff could be enormous. Well, that's an understatement from my perspective, and possibly yours as well. Many people with autoimmune issues take immunosuppressive drugs that dampen the entire immune system, making them more vulnerable to infections and cancer. But a vaccine that tamps down the immune response to a specific antigen wouldn't have that effect. It's a field where a lot of people want to make the breakthrough and become the next chapter in the glorious history of vaccinations, Steinman says. And as far as glorious history goes, I'd settle for a glorious history in multiple sclerosis. I guess I'm being a little selfish there, but we have waited a long time for something that's promising. And let's just keep our eye on this one, because who knows, it might very well be the next big thing. Well, there you have it. Very interesting stuff. I've emailed someone that I respect to find out whether they have heard about this and what they think. And here's his reply. Yes, I am aware of the work. Unfortunately, it is an animal model with a well-defined autoantigen. We are still looking for the autoantigen driving MS. Looks suitable for autoimmune disease were we to know the antigen. And that's Gavin Giovannoni, the neurologist who talks all about smoldering MS. You've heard me talk about him many times. And of course, his point is well taken. This research probably represents a step toward getting a vaccine for MS, but as Professor G says, we have to get the right antigen that we're attacking, and we don't at this point know that antigen. So things are still way off in the future. All we can really do is watch and wait. So I suggest that that's what we all do, and that we do it together, because, it makes the waiting go a lot faster when you have friends to wait with. And in the meantime, while we're waiting, do take really good care of yourself. And I'll see you again in my next video.